Good evening, everyone. So in my 10 minutes, I'm going to try to answer the question, what is a neuron and how is it made, by looking at the evolutionary origins of neurotransmitters. All right, so the human brain is the most complex organ in the human body. It allows us to do amazing things like create revolutionary inventions and beautiful works of art. The human brain is composed of 4 lobes, the cerebellum and the brainstem, and also 86 billion neurons. One single neuron can have an average of 10,000 connections to neighboring cells. And these neurons can use multiple signaling molecules, including neurotransmitters, to communicate with these neighboring cells. So in short, the human brain is extremely complex. And so in my thesis, I wanted to look at where this complexity came from. So since neurons allow the brain to communicate with the rest of the body, I wanted to look at how do neurons evolve. And so to do this, I'm using a model organism, Aplysia californica, aka the sea hare. So this is a mollusk, so it's related to snails, slugs, and also octopus. So you can see it here inking purple. But that's not why I find it fascinating. I find it interesting because instead of 86 billion neurons, it only has 10,000 neurons in its nervous system. And some of these neurons are large enough to be seen with the naked eye, like the one shown here on a US penny. So this is the sea hare nervous system versus the human brain. As you can see, morphologically, they're very different. But what they have in common is that they both use neurotransmitters. So they use dopamine and serotonin just like we use dopamine and serotonin. So serotonin is involved in a lot of different functions. So it's involved in mood, anxiety, addiction, obsession and compulsion. A lack of serotonin can lead to depression, and in mollusks and also in arthropods like lobsters, food sensing. Dopamine is involved in some of the same functions, so mood, anxiety, addiction, but also in alertness, energy, arousal, and most importantly, paying attention, which I hope you're all doing right now. All right, so since we've gone through a little bit how these neurotransmitters, what they do, now we're going to go how they work. All right, so you have 2 neurons, neuron 1, neuron 2. When the cell is at rest, we have these little storage bubbles called vesicles, and inside is neurotransmitter. So that when the cell gets excited, these vesicles move to the edge, they bind with the membrane, and they get dumped into this gap between the 2 cells called the synapse. If enough neurotransmitter gets dumped, then the second cell also gets excited and fires its neurotransmitter onto the next neuron in the train, and so on and so forth. And that's how signals get communicated through neurons in the brain. So what exactly does this first cell need to be considered a serotonin neuron? So first, it needs to be able to make the neurotransmitter. Then it needs to be able to put it into the little storage vesicles. And finally, after that action has happened, they need to be able to clean up the excess neurotransmitter and recycle it so that the whole thing can happen again without oversaturating the cells. And so to do all this, you need 5 genes to be turned on at the exact same time. So they need to have coordinated expression in the exact same cell. All right, so my real question is, do these 5 genes that help us make serotonin get turned on the same way in the sea hare as they do in people? Or do we have more than one way to make a neurotransmitter? All right, so looking at the sea hare is actually the final piece of the puzzle. All of these animals that you see here have a left and a right, so they have 2 axes of symmetry, they're bilaterians. And using the sea hare fills in this last missing branch with a representative organism. And so my research could go one of 2 ways. We could find that the sea hare makes dopamine in a new way, turns on these genes in a new way. And so that would be biomedically relevant, because then we could use it to synthesize dopamine in a new way and potentially treat dopamine-based diseases such as Parkinson's. If it's made in the same way, then this answers an evolutionary question, and it suggests that the common ancestor of all of these animals probably had dopamine. And so this gives us kind of an idea when the complex brain began to form into what it is today. And so the same 2 principles are true when we look at serotonin. The literature looks more at dopamine, but in the sea hare, it's a lot easier to look at serotonin because we have 2 giant serotonin neurons. Yes, they're large enough to be seen with the naked eye, and they're very great to work with. So I mentioned before that one of the important things is coordinating gene expression. And my lab does this, or looks at this, by looking at genomics. So here we have the human genome. This is written in DNA, and it encodes every possible thing you can need to make any sort of human cell. But you don't use the whole thing in every single cell, though you have a full copy in every cell. So in order to express a gene, you go from DNA to RNA to protein. That's because your construction crew are ribosomes. They don't speak DNA. They only speak RNA. So you have to go into the instruction manual, pick whatever instructions that you want, and photocopy it out and transcribe it so that the ribosomes can read it. They say, OK, great. They read the instructions. They build the protein. Then they throw it away. So you have to photocopy the same genes over and over again to get them to be consistently turned on. So how can you quickly get the same genes to turn on? Post-its. So these post-its are transcription factors. They're a special code, and they mark what genes are supposed to be consistently turned on in a cell. So if this is a nerve cell and this is a skin cell, you have a different post-it code. And so what I'm trying to figure out is I'm looking at the genome of the sea hare, which is, luckily, a third of the size of the human genome. So a little bit easier to work with, but the same sort of post-it code. And so I'm trying to figure out, do the transcription factors that turn on the 5 genes to make serotonin, are they the same in the sea hare and in people? And so to do this, I'm actually losing my clicker. I actually do a lot of genomic sequencing and transcriptomic sequencing. So what I did was I isolated individual serotonin neurons and individual neurons of other that have different neurotransmitters. And so I gathered all of the RNA together, so everything that's been photocopied out of the cell, and this is what we call a transcriptome. And so we take all this genetic material and we load it onto a chip like this. Yes, these are the most expensive earrings that I own. And we put it onto a genomic sequencer. A lot of data comes out. A lot of boring computer stuff happens. But basically what I ended up with is a short list of potential post-it notes or transcription factors. And one of those is PAX6. So in other organisms, PAX6 is involved in eye development. If you have a messed up copy of this gene, you're either going to get partial eye development or no eye development at all if both of your copies are messed up. But in the sea hare, it's not involved in eyes at all. It's involved only in serotonin neurons. So here it is labeling my 2 giant MCC cells and also some other serotonin cells, but only serotonin neurons. And so the same post-it note that has a different role in the human genome is now regulating serotonin neurons in the sea hare. And so basically what my research suggests thus far is that we have a new way to recruit the serotonin genes and make a serotonin neuron, which is really quite exciting. So I'm a 4th-year graduate student, and I'm still working on some other candidate genes to double-check this, but right now my boss is really excited too. So with that, I'd like to thank my lab. This is my boss here in the wetsuit, because yes, we are sequencing on a boat. And this is the rest of our lab. I'd also like to thank the Whitney lab in St. Augustine and my coworkers, and go Gators. All right, thank you very much.